Welcome friends. This video is about the regulation of glycogen metabolism. This is the coordinated and reciprocal regulation of both the glycogen breakdown and glycogen synthesis. These glycogen enzymes are regulated allosterically and covalently. First we would discuss the allosteric regulation. Let's discuss what is the function of this glycogen phosphorylase. It simply breaks down what? Glycogen. On the other hand, this GSA, that is glycogen synthase, makes glycogen. Because this is coordinated control, we are discussing the enzymes of both these pathways together. And this is allosteric regulation first. If this is breaking down glycogen, what it would make? Glucose 1-phosphate and then to glucose 6-phosphate, which is entering into glycolysis. And in this case, it is adding glucose to glycogen chain. What kind of molecules would inhibit or activate these enzymes? This enzyme would be active when? What is the function of this enzyme? It breaks glycogen. Why it breaks glycogen? So that we can produce glucose. For what purpose? To continue glycolysis so that we can produce energy. So this is active during low energy states. During low energy states, because we want energy. So high AMP stimulates this and low ATP would activate this enzyme. On the other hand, this glycogen synthase, when it would be active? Opposite of this, when we do not want energy, we want energy to be stored. So high energy states it would be active. Its inactivity, when it would be inactive? When there is a lot of energy available. So we do not want that energy, because the function of this enzyme is to convert glycogen to glucose units to produce energy, but energy is already available. Why would we want this enzyme to be active? So it is inactive during high energy states, or we can say high ATP and low AMP. And if we see in this case, this is inactive during low energy states. Why so? Because there is no energy available. Why would we want to convert glucose to glycogen when there is no glucose? So this is inactive. So we can say high AMP and low ATP is stimulating this enzyme, and high ATP and low AMP is deactivating the enzyme. In this case it is opposite, activating, and here it is deactivating. So this makes sense, isn't it? What we can say more is that if this is happening, what is the purpose here? To make energy, and in this case to store energy. If energy is more, we are going to store the energy in the form of glycogen, and if energy is less, we are going to convert glycogen into energy through glycolysis. So now let's talk about the regulation of the breakdown enzyme glycogen phosphorylase. This is the covalent regulation which is carried out using the hormones glucagon and epinephrine. So the hormones come into play now. Before we discuss this mechanism, this pathway, I want to tell you something about glycogen phosphorylase. This enzyme exists in two forms, that is glycogen phosphorylase A and B. This is active and this one is inactive. We want this enzyme to get activated. That is what we are going to discuss in this pathway. This pathway is an enzyme catalyzed cascade. One enzyme is going to activate another enzyme, and that activated enzyme would activate another enzyme. So these hormones are coming from two sources. One is liver, these hepatocyte cells, and myocyte for muscles. Epinephrine and glucagon both are released from hepatocyte. In case of muscles, it releases only epinephrine. One more thing it releases is calcium ions. We want glycogen to be converted when there is a shortage of glucose. In case of muscles, when we are doing vigorous exercise. Okay, so in that case we need glucose, and glucose is provided with the help of this enzyme glycogen phosphorylase that would convert glycogen. So what we are doing here is this epinephrine and glucagon, they are going to act on an enzyme. That enzyme is adenylate cyclase. Before they act on it, before they bind to it, this is in its inactive form. It has 3 units. I can draw like this: alpha, beta and gamma. In its inactive form, it is bound to GDP. When these hormones bind, what happens? These 2 units, gamma and beta, they are gone, beta and gamma. Only one unit is remaining, that is alpha with GTP now. So this is activated, G alpha, after these hormones bind. Active adenylate cyclase would convert ATP into cyclic AMP. So now this is increased. Cyclic AMP is increased. This increased CAMP going to activate another enzyme which is known as pKa, that is protein kinase A. So this is kinase. Kinase means it is going to phosphorylate something. Active pKa is going to phosphorylate inactive phosphorylase B kinase. This is another enzyme which is kinase but in inactive form, activated by pKa. This enzyme, when gets activated, is going to activate another enzyme which is what we are interested in, that is glycogen phosphorylase in its B form, inactivated form, gets activated with the help of phosphorylase B kinase. Okay, this phosphorylase B kinase in turn we can say activated by one thing more, that is calcium in case of muscles. When there is increase in the concentration of calcium ions during, we can say, heavy work, this is activated. So this is now activated, glycogen phosphorylase A. Now it's in A form after its stimulation or activation. This can act upon glycogen and would convert glycogen into glucose 1-phosphate, and the fate of this glucose 1-phosphate can be in liver. In case of liver, it would enter into blood to maintain the blood glucose level, and in case of these myocytes in muscles, it would undergo glycolysis to give energy, that is for muscle contraction. So now we have discussed the effect of glucagon and epinephrine during low glucose level. What would happen when there is plenty of glucose available after a carbohydrate rich meal? At that moment, we would want glucose to be stored as glycogen. In this case, another hormone is coming into play, that is insulin. This is going to signal the enzyme glycogen synthase to get active and store glycogen. make glycogen or store glycogen. Like phosphorylase, this glycogen synthase also exists in two forms, that is active A form and inactive B form. In case of phosphorylase, the phosphorylation was bringing about the activation and dephosphorylation was bringing about deactivation. Here it's opposite. Phosphorylation of glycogen synthase is inactivating this enzyme, and dephosphorylation of GSAs is activating the enzyme. So let's see here. We have this glycogen synthase which is in the active form. It has serine residues present on its active site. There are so many serine residues, and the enzyme which is in this red color enzyme, glycogen synthase kinase 3, is going to phosphorylate at these residues. So this is phosphorylated, and after this phosphorylation, this is inactive. Now this inactive form cannot synthesize glycogen, but we have a lot of glycogen, a lot of glucose available, so we want that glucose to be converted into glycogen. That this effect is reversed, this phosphorylation is reversed with the help of another enzyme here, PP1, that is phosphoprotein phosphatase 1, that would remove these phosphate groups which are on the serine residues and cause activation. Now we can talk about the effect of glucagon and insulin and the molecule glucose 6-phosphate on this enzyme phosphoprotein phosphatase. If glucagon is there or epinephrine is there, what does that mean? We want glycogen to be converted into glucose. We need energy. But this enzyme is responsible for activation of glycogen synthase, means we want glycogen to be stored, convert glucose to glycogen. So this is opposite, this is going to inhibit PPI. Okay, insulin is released in response to more blood glucose. So store, active, so this is going to activate GS synthase, glycogen synthase. So this is activating here. In case of glucose 6-phosphate, we have more glucose or more glucose 6-phosphate activating the enzyme.